Good day, everybody. We are from Group 5 presenting about Case Study 5. These are our group members. In this presentation, we'll be talking about three common groups of diagnosis method, which are indirect diagnosis, direct diagnosis, and serology diagnosis, as well as how is the antibody being measured in Case Study 5. The virological related techniques used are grouped as follows. We'll be starting with virus isolation.
The very first virological related technique used in this case is an indirect diagnosis skill, which is the virus isolation. We take the virus out from an infected host and then propagate it in cell culture. In this case, the NDV isolates are replicated using 10-day-old embryonic egg, and then the virus will grow in allantoic membrane. Moving on to the procedure of virus isolation, the first step is to place the egg in front of a light source and then locate non-vein portion. Next, make a pencil mark on the area, drill a hole at the top of the egg, and then by using a tuberculin syringe, inoculate the egg. Pass the needle through the hole in the shell and then inject the inoculum. Then incubate the eggs for 40 hours at 37 degrees Celsius. And lastly, the virus replicates in the cells throughout the incubation period. After inoculation of virus in embryonic egg, indication of virus growth can be confirmed by observing cytopathic effects such as vacuolation, syncytial formation, and so on. Next, the embryonic egg can be directly observed. There will be evidence of apoptosis and necrosis of embryo and many more. For further confirmation, allantoic fluid can be used for HI test. For NDV, it will agglutinate the RBC.
Subsequently, elution test is used to differentiate strands of NDV by the indicator of elution time. What is elution test? Elution of NDV is the process of extracting NDV from red blood cells by controlling virus multiplication via pH and temperature adjustment. Elution time of NDV is the time between the start of the elution and the time at which all the NDV elutes. Different strands of NDV perform different elution time. Let's look. The elution time here means the time taken for the NDV from the well of hemagglutination endpoint in HA test to elute. The mean elution time of velogenic strand is 118 minutes, mesogenic strand 59 minutes, lentogenic strand 25 minutes.
Next, let's talk about RT-PCR, which is a direct method. Real-time reverse transcription polymerase chain reaction is used for sensitive and quicker identification of RNA virus, which is applied to detect the virus genome of NDV. From this, we can obtain quantitative results. Before doing RT-PCR, we need to obtain the samples from swab techniques. The first swab technique used was cloacal swab. Insert the entire tip of swab into the cloaca and swab with 2 to 4 circular motions while applying gentle pressure against the mucosal surfaces, and then shake off any large residues and then put it in the tube. The second method used was oropharyngeal swab. Gently roll the swab tip around the inside of the bird's mouth and behind the tongue and then place it into the tube.
Now let's briefly look through the procedure of RT-PCR. Firstly, prepare the cloaca and oropharynx swab. Next, carry out RNA extraction. Third step, decide if you want to use the 1-step or 2-step RT-PCR technique. If using the 2-step technique, reverse transcription is carried out first. Then, reagents are added into the PCR tube and incubated in a thermocycler. Step 6, once reverse transcription is done, we will proceed with PCR. Step 7, the first important process in PCR is denaturation, where double-stranded DNA is denatured to become 2 sequence. Step 8, annealing process then occurs where DNA primers will anneal to target virus sequence. Step 9, elongation process takes place to synthesize the subsequent sequence, and the cycle is repeated to form many copies of DNA genome. Lastly, step 10, gel electrophoresis is done to record results.
The result is positive when the DNA copies pass the threshold value, so all of these samples above are positive while the green line is negative. At the point where they cross the threshold is their CT value. A lower CT value will indicate a higher amount of viral RNA in the sample.
Next, I'll be presenting about the serology approach, which is the HI test, hemagglutination inhibition test. So this test have the main aim is to detect the antibodies to paramyxovirus type 1, which is the virus of Newcastle disease. So the principle is that the HI test is based on the NDV antibodies' ability to inhibit the RBC agglutination, and this is done by the hemagglutinine on the viral envelope of virus. So the virus have two surface glycoprotein, which is the HN and F protein only. So the HN protein has the ability to agglutinate the RBC in the absence of antibody. So if it shows diffuse red color at the bottom of the well, the result is negative. And if there is a presence of antibody which hemagglutination is inhibited, you can see a small pellet or button of RBC at the bottom of the well. This means that the test is positive. And if the animal has not been vaccinated, but there are antibody detected present against the virus, this indicates that animal is infected by the virus. So the other purposes of HI test is to measure the immune responses of the poultry, and this is done by assessing the level of antibodies presented via the HI titer. When there are virus isolation that showed presence of hemagglutinating activity in the current electrolyte fluid in egg, this may indicate that NDV virus is present. Hence, HI test can be used for further confirmation.
Next, we go to how is the antibody measured. We use hemagglutination inhibition test to measure the antibody in this experiment. So we use the hemagglutination principle where the hemagglutinine neuraminidase or HN from the Newcastle disease virus NDV to bind to the sialic acid receptor on the red blood cells RBCs to form the lattice formation. So basically in hemagglutinine inhibition test, the serum of the Newcastle disease virus infected animal is mixed with the RBC and Newcastle disease virus itself, and there will be no agglutination of the RBC. So basically for the positive results, the antibodies in the serum react with Newcastle disease virus and neutralize them. Where the HN is occupied, cannot agglutinate the red blood cells. Where the negative results, there is no antibody against the HN, thus the HN agglutinate the RBCs. For the procedure, firstly, we must obtain a preparation of the Newcastle disease virus with known HA titer. And the second one is prepare 2-fold dilution of tested animal serum to be tested. 1:2 ratio, which is 1 log 2 to 1:512 ratio, which is the 9 log 2. And for the third step, we add a fixed amount of NDV to every well of 69 well plate. And for the fourth step, allow the plate to stand at room temperature for 60 minutes. And for the fifth step, add chicken RBC and incubate at 4 degrees Celsius for 30 minutes. And lastly, we read the wells. So for the result, for the positive result, the RBCs sink to the bottom and form a button formation, which is like this. And for the negative result, RBCs attached to the NDV form a lattice formation and will appear such this picture. For hemagglutinine inhibition titer of the serum, the highest dilution of the serum antibody that prevent hemagglutination. So in this case study, 48 birds serum tested show button formation until the last well with HI titer ranging from 2 to 256, and 30% of vaccinated chicken serum contains antibody that are able to block hemagglutination from occurring at HI titer of 8.
This is the page of references, and it marks the end of our presentation. Thank you.